All right, so we got two. There's a PowerPoint that will go over introduction to metagenomics. What are we talking about when we talk about metagenomics? And then also get us kicked off with QIIME. And then there's a collab notebook that is super ugly and kind of broken, but I'm giving it to you anyway. QIIME is, over the last week, rapidly climbed the list of my least favorite software, but it's what all the metagenomics people use. So, if you're a data science, computer science, undergrad or grad student, looking for a software platform to improve, a lot of people use this. They have the stats right on their webpage. So, anyway, first thing we're going to do is look at the PowerPoint. How many of you are in a lab that does metagenomics? Just curious. One. Whose lab are you in? Buckner.
For my project at least.
Okay. No Ravenscraft, Chang lab. Yeah? Dr. Chang? Yeah. Those are the two that, so Dr. Ravenscraft does insect gut microbiomes. Dr. Chang's lab does both soil and human gut microbiomes. I guess when it's soil, it's more metagenomes, but in any case, what are we talking about? What's metagenomics? See if I can just skip the whole intro. You already know.
So you're getting info on all the different genomes of organisms in your sample.
Yeah, it is. It's not Mark Zuckerberg genomics. Nobody gets that joke? Meta? Oh, man. And I thought I needed caffeine. Or maybe I had too much caffeine. Okay, studying what lives on and in us. My favorite example of metagenomics when it was first becoming a thing is some of the guys that, well, the PI that designed Galaxy, who many of you have used, the server. They went on vacation on this road trip through Pennsylvania, and they had a new genome center at Penn State, and he just decided to scrape his windshield and sequence it to see what all the bugs he'd hit that were splattered all over his windshield, literally. And they got a paper out of that, like environmental sampling. But exactly right. The sample is somewhat arbitrary. Sequence dirt, sequence ocean water, sequence air nowadays. And it's mainly to find out what are the microbes that are in that sample. Ultimately, that is the sort of biological question behind this. What is in that sample that has DNA? Three main approaches. So the kinds of data that you're most likely to see if you're doing a metagenomics, 16S, ribosomal DNA sequencing, is probably the most common because the 16S is so conserved across microbes, and it's good for identification at least to the level of genus, maybe species. And because it's good to that level, it provides a good measure of diversity. And part of it ultimately depends on I guess part of whether you do 16S or whether you really need to do one of these other more deeper sequencing or naive approaches to what you're sequencing is ultimately the question. Do you need to know what the function of those microbes is in whatever your sample is? If you're doing dirt or ocean water, some kind of environmental sampling like that, sometimes you're not so interested in the functional qualities of that community, like are they metabolizing X, Y, Z. Whereas if you're doing human microbiome, for example, you might actually be interested in what's the function of the main components in the microbial community. The alternatives, 16S, I would say, like that's a targeted approach. You're doing a known conserved gene. You can do deep sequencing of total microbial DNA. It's a more comprehensive view. You can see the variation within species, so you can get at a variation at a lower level. Potentially detect things like horizontal gene transfers, which we know are important. One of the bioinformaticians that we had phone interviews with on Tuesday, I mean, he's making a whole career out of detecting plasmids in metagenomic samples, stuff that is not attached to chromosomes. And then your level of species sampling, how much strain variation you can detect. If you're interested in that granularity of analysis, then it will depend on how deep do you sequence, and do you sequence regions other than 16S, which is more conserved. And then you could do RNA-seq. This will at least tell you about all the proteins and potentially give you a better functional indication of what the microbes are doing in that sample, right? Because even in a relatively reduced microbial genome, not all genes are expressed in all samples, right? So we know there are lots of microbes that are on or in us pretty regularly that don't cause any problems until they get stressed. And then they start to produce toxins. The cholera bacteria is the poster child for this. It's pretty common in the environment, but it doesn't really start producing toxin until it gets stressed. Common myth, you still hear this even in our research talks, that humans are vastly outnumbered by the number of microbes on and in them. That's probably not true. At least, but there, as a ballpark, there are probably roughly as many microbes living on and in you, on a per cell level, that is. And if you're interested in that, especially if you're doing metagenomes or human microbiome stuff, you might be interested to go look at that paper. I put this in here just because it's such a pervasive myth. It was around for a while, and it wasn't until we had good microbiome sequencing where we could get better ideas of how many microbes there are that it's probably not true. It's also a good lesson in taking numbers at face value that are just dogma in the literature. The human-chimp similarity was one that was around for a long time, like we're only 1% to 2% different than chimp on a nucleotide level. Another myth that I personally helped bust with that whole gene family evolution stuff. That's only on the stuff that's highly alignable that we're 1% to 2% different.
How different are we then?
Depends on how you count.
How do you count?
Well, the problem is we both have roughly the same size genome, but our chromosome composition is different. And you can't align all the genes very well, and you can't align the intergenic regions very well. And so I don't know off the top of my head how many base pairs differ, because you can't align a bunch of those regions. Like, you don't know how they align.
But for like, I can ask you later. We don't need to go down.
Yeah. It's not a trivial thing to count, is the bottom line there. When you ask how different are two things, you can have a total genome size answer. You can have a alignable protein coding sequence answer. Those are usually the two general metrics. Then you have structure, how different are they structurally. Website, again, this is human microbiome focused. I think this also has mouse in it maybe, or at least link outs to mouse. But just in terms of I don't know why I ended up so human focused, probably because most people are interested in human, especially if you're coming from data science, because this is where most of the research is done. The thing that gets focused most often, I should have done this as a quiz question, is feces. That's the most common. That's the data that you're going to analyze today. Well, some of it. Yeah, feces and mouth. Those are the two that most often get sequenced. This is actually probably a year old, so the numbers of studies are going way up. The thing that's sort of nice about this, and another reason that I kept these, this is probably one of the widest databases. So if you're interested in, say, doing a human microbiome project for your semester project, this is where you can go get data. If you look at what do we know, what are we finding out, I don't know if this is surprising or not. It varies depending on where on the body you sample. It varies across populations. They're both environmental and genetic components to this. And there is also ontogenetic shifts. Sort of interestingly, the y-axis here is how different is, and the colors are different populations: urban American, rural community from Malawi, and rural Native Americans. And the y-axis is how similar is an infant to an adult of that population. And so you can see that in all populations, infants are quite different than the adults. And then as you get to like 3, 4, 5 years old, it basically levels off to where you've basically stabilized, and roughly child at that age to an adult at that age from that population. A child at the given age here versus an adult in that population. And one of the take-homes there is that gut microbiomes are somewhat population-specific, somewhat environment-specific, but they often stabilize at a pretty early age. And one of the interesting things, and this is not only coming out of the human literature, but I think one of the cooler areas of research in metagenomics is the interaction between the host genetics and the microbial community that they all ultimately stabilize on. There seems to be strong genetic component to your microbial community, even given similar diets, similar environment, stuff like that. This is a same kind of Manhattan plot, same kind of, you could think of this analogous to the EQTL stuff we did. This is a genome-wide association study for where microbial community is the thing you're trying to predict, and then you've got the SNPs there. So you have various SNPs that are associated with the frequency of, I guess these are mostly at the genus level. There's some that are at the level of order and family, but loci in the human genome that predict composition of microbes. Questions about that? Does that make sense? Okay, just a little bit more background that comes up when we talk about what we're measuring, right? Because the question is often about the diversity, more so than the specific species, unless you have a known function that you're looking for. I had these slides already from teaching ecology, so that's why it switches to forest instead of microbes. But the diversity metrics are the same. Alpha diversity is typically measured in terms of species richness or the relative abundance of species. So this cartoon example of two forests, the question you might ask is, which of those two forests is more diverse? What's your instinct? The community one? Yeah, well, yes, on some level, like if you're walking around in that forest, it's going to feel more diverse because you have a more even distribution of species in that one. There are the same number of species in this one, so don't need to walk through computing these things for you guys. But right, this, by the Shannon Diversity Index, which is probably the most common one for alpha diversity, has a much higher level. It's just, basically giving you a metric to describe this evenness of species composition in that forest. The other major difference, it's kind of a different question, is beta diversity, which is asking about specifically what's the difference in diversity between two communities. So even though I asked which of those two do you think is more diverse, the Shannon, that alpha diversity, is just telling you about the diversity within that forest. So this is telling you that the alpha diversity by the Shannon metric is 1.3 in this forest and 0.7 in community 2. And so you would say the alpha diversity is higher in community 1. Yeah.
So we did the same things for, I guess, in my
These are undergrad slides, yeah.
Yeah, I was a wildlife major. I do have a question. Does this test index cover also species name? Does it cover how high? Like the crown of itself?
So species name versus,
Like, does it cover like the general descriptions of each tree, or just the species itself?
oh, this would just be the species names. This index, though, isn't, you're not restricted to using species names as long as your metric is in this context, at the same taxonomic level. Like you could do it for orders, and we'll see in the metagenome stuff when we run through the collab notebook, that does become relevant for the microbes.
Like this by hand.
Especially if lots of your microbe sequence can't be identified all the way to species level, then probably not a good idea to do your diversity metrics at the species level, because you get a biased sample that way, or a biased index that way. This question about whether they're different between community 1 and community 2 is called beta diversity. The two most common metrics are Bray-Curtis and Jacquard. Bray-Curtis is abundance-based, sort of that instinct that you had before about the evenness, like are they equally even between the two? Jacquard is based on presence-absence only. So Jacquard would be, well, actually I think I've got it on, yeah. Again, not going to necessarily walk through the calculations here, but you know, if you want to take the time, you can walk through. It's pretty simple calculations for all of these. You're just summing across the difference in species between, say, population 1 and population 2, or X and Y, over the total number of species in each one. This, like a Bray-Curtis of 0.5, maybe you get this instinctively. You've got both species in there, so these are not maximally different, but I would say a Bray-Curtis of 0.5 is moderately different. Jacquard's just based on presence absence. In this case, they're both, all the species are present in both, so your Jacquard distance here would be 0. And again, I'm putting these in just so you have in mind what they measure, because they're referenced in the metagenomics literature and in QIIME's outputs, so give you an idea of what you're looking at. Just thinking about in the big picture of what we've been doing, why did I put single-cell RNA-seq and metagenomics here, right? The cells were the unit of data in the single-cell RNA-seq. Here we've got communities of microbes, and we were trying to predict cell types in single-cell. Here we're trying to predict species from our samples, or some taxonomic level. We're trying to identify microbes. Our feature table, before we had the genes as our rows with the number of reads, or the normalized number of reads for that gene in each sample. Here we've got, QIIME calls them ASVs, amplicon sequence variant. I bolded that because if you go read literature from more than a few years ago, you see OTU all the time. The operational taxonomic unit. So yeah. Right, so this is one where this shift has basically occurred as the sequencing depth has gotten better. The central problem is often that you don't know whether a read with, say, 1 or 2, for example, differences belongs to the same strain, or that's a sequencing error. And so until a few years ago, they would just set a similarity threshold at, say, 97%. That's the most commonly used one. I think it used to be default in QIIME, which is why it's the most commonly used one. And now they just take a different approach. So there's a denoise step. This software data tries to correct all the sequencing errors first, and then keeps all the strain information. So every difference gets its own amplicon sequence variant. So it does clustering based on sequence similarity, tries to correct the sequencing errors based on that, and then anything that's not a sequencing error but still has a difference gets its own strain identification, or ASV. I just flew past, like, you do the clustering, this dimensionality reduction that we did with single cell. Same kind of thing is going on. I wanted to highlight the fact that PCA and PCOA are not the same thing. Give you a little table there. Not going to work through PCOA in the same depth like we did with PCA. The main reason that you use PCOA in metagenomics is that you're already computing the distance matrix for sequence similarity when you do the denoising. And so this coordinate axes are more straightforward, and the data is better suited to this method. Yeah, I'm going to leave it at that. I already, like, the whole thing about trying to correct sequencing errors so that you get strain information is the objective, which isn't achieved as straightforward a way with a PCA, which just uses the variance. Like, how you measure that variance would be not clear, at least not clear to me. I know I explained that as clear as mud, but I'm going to move on anyway. I don't want to run out of time. The connection to machine learning here is that we're going to end up with this table that's got all of our sequences that we've identified as separate strains, and then that's very highly dimensional, and what we really want to know is how many of those correspond to taxonomic units. And so that's where the clustering and ordination happen, and then you put taxonomic labels on that based on its match to a known data set of microbes, or they are left unlabeled, right? I've mentioned a couple times, like, often you can't identify all the way to species, or even, you'll see there are some that are left at the order level, for example. And that's because there's a lot of microbial stuff out there that we just don't know what it is. Okay? It just doesn't match to something known. So we're going to jump to QIIME 2. This is a link to the main website. And why use a built-in software package? Because it's got those databases of known taxonomy. So if you think back to the cell type analogy, I kind of skipped over this in a previous slide. The labels, right? How did we get labels in single-cell RNA-seq? Or what were the labels called in single-cell RNA-seq? Well, so what is the classifier using to call a cell type? How do we know that, this cluster of cells are B cells or T cells or fibroblasts? What are the labels in the single-cell RNA-seq? So clustering in single cell, how did you do what's the clustering based on? Single cell RNA-seq. What's the clustering based on? Gene expression, yes. So you've got all the genes that are expressed in a cell, in all the cells, you cluster based on that gene expression profile, okay? So now you've got clusters. I've got a group of red dots and a group of orange dots. How am I going to try to figure out what does that red group and that orange group mean? What are the labels I get to pull in to try to help me understand what are those clusters? No.
Existing annotated data of cell types, like what each cell type's expression looks like in reality?
Yeah, so marker genes, right? Marker genes. So things that are, you look at what's the most highly expressed in this red group that's not expressed in any of these other groups, and try to, is that a marker gene? Or you can go the other way too. You've got a database, you know that B cells, T cells, fibroblasts, they have specific marker genes. You look at your clusters and say, which ones have the high expression of the marker gene for that cell type? Okay, that's how you go from unsupervised learning, that's the clustering, to then applying some kind of supervised approach, because now you've labeled those clusters. Then you've got a classifier that can label new data. So the translation for metagenomics, right, is that we're going to have species names. So cluster, the data going into QIIME are just the reads. So we've got genomic reads instead of RNA-seq reads. And so we're clustering the reads based on how similar is the sequence. So you get a red cluster and an orange cluster, and that tells you something about, each of those dots is a read now, and we want to know what it came from. So this is the step where for single cell we used caffeine's wearing off. Marker genes, right? So in single cell we got marker genes. So in metagenomics we've got our clusters. What are we going to do? How are we going to label our clusters?
Compared to a known data set, like groups.
Yes. Yeah. Right. So BLAST, or in this case, it depends. Like BLAST, hidden Markov models, something that will be able to take the labels from a known data set where you know the taxon ID and assign it to your clusters. And that's where QIIME comes in. That's the main benefit of using QIIME. Okay. Okay. because it has the databases, it knows how to search, and it's got all the, the tools for doing the clusterings and things like that. We could do all of that other stuff ourselves. We know how to do PCA. We can use scikit-learn to do a lot of those steps, the unsupervised clustering and stuff like that. But QIIME has it built in, and it has the database to give us our labels. So that's why we'll stay in that environment. Okay, so I've kind of walked through that slide, the workflow. And you're going to do that in the notebook and the tutorial anyway. This is just a graphical representation of that. You're sampling some, in this case, gut, could be soil, doesn't really matter. You process the data, do quality control, denoising, and then you get into what most of the tutorial is. How do you assemble those, label them with some taxonomy, make some functional inferences about what's happening in the microbiome. And these are the kinds of questions, kinds of research areas that of people who use QIIME who are doing these metagenomic or microbiome studies. So this is the tutorial. I've given you that link. So this is what you're working through. And you should walk through this on your own. But the reason I gave you even as sort of ugly and non-functional as some steps at the end are, this is a few days of me trying to make it work. And so QIIME, the thing that makes QIIME very unfriendly is that it's not one group developing it. So the different modules get updated at different rates. This is my take on what's happening. And so way back at the beginning, I talked about how Python is bad for just dependency hell. QIIME seems to always be in dependency hell. There's also a little bit of complication just because I'm sort of forcing it to run in Colab. I started to move on to Bridges 2, and it wasn't any easier, so I gave up right away. That's all fair warning and explanation of this will run okay for a while. When you get down to boots, it won't work. But I left it as a non-working boots install. And you can collapse that and do taxonomic analysis, and it will skip right past that. So there's a couple steps in here that take a few minutes, but we'll hang around for the rest of the time, let you guys start walking through this. The other thing about the tutorial is if a step fails, for some of these, you can download the intermediate data set. So pretend it worked and put that in your Colab notebook, and then it will just pick up from there because you'll have the outputs. Yeah? That whole time? Yeah. There we go. Yeah, when I hit the link, it didn't move over. So this is the gut to soil axis tutorial. That's interesting. I'm wondering right now if on the recording, on the video, if it's showing that or this. I think it's showing this. So on the recording, you'll see what I was pointing at, even though in class you could not. what I was pointing at is that if there's a cell that doesn't work, right, you can walk along and hopefully you'll be able to see, even though my cells don't look exactly like this because I've got extra bash commands in there to make Colab run this bash micro, I forget it's mini or microconda environment. That's what I mean by it being ugly as a Colab notebook. I just didn't go back to make aliases to clean it up. But you'll be able to see as you walk through this, and if a cell doesn't work, you know, try to make it work, but if not, there are links here to circumvent the dysfunction and get the data file that you need, the intermediate outputs. Okay, there are caveats to everything with QIIME. Some of these, actually many of these, like come down here where it starts getting broken, and oh, that one. That's data 2. These might work. Some of these download ones are broken. That's what I was going to show. But so mine has finished the install. So that second cell should install everything you need. And then after that, most of the cells are pretty fast. One thing that I skipped over in the benefits of QIIME, and understandably, much like lots of genomics people have their students, well, actually, it's not just the students. People use Galaxy because it makes repeatability easy, right? It builds the pipeline as you go. QIIME has this format where everything is an artifact or visualization, QZV, QZA file. That's I don't know if it's the server name. I don't know where it came from, but the name of this format they call provenance, and it keeps all the metadata in the same file with the data. That's why these QZA, QZV, these are unique to QIIME. It's the only thing I know that uses these. It has a tool to convert them to HTML if you want to show them in Colab or something like that. But what that means is the only way to look at them is to use their server. So you just Google QIIME view, and you have to drag those files into that in order to look at them. Once you actually get some data. Okay, so like the first one that you'll generate is this sample metadata QZV. So to look at that, I would download it and go to my, come on, my QIIME viewer page. Put that sample metadata in there. And so this is the file that I just generated in Colab, downloaded it, and then stuck it in the QIIME viewer. And this is just the sample info. So maybe you could glean that from the file name. It's the sample metadata. But it's basically telling you what kind of sample, and it has a sample ID. So next thing will be to go get the actual sequence data. Demux is a name has nothing to do with my last name, even though it's very close. It's about demultiplexing. Just fortunate or the reason I bring that up, it's been confused in a few experiments and collaborations that I've had before. People, thought I made a typo or thought somebody made a typo naming the file after me.
All press is good press.
Yeah, right. Except not when somebody else's grad student is banging their head against the wall. This data came from the Damath lab. Why do they call it Demux? And it doesn't just happen with metagenome data. Like, it's any demultiplexing. In the stuff we've done so far, we've kind of ignored the demultiplexing part. Often that happens on the Illumina machine, and so you don't have to deal with it at all. So that one takes, I think, actually it didn't, that's good. So now we can look at our data. We've got a Demux QZV. Going back to the QIIME viewer and putting that Demux QZV. And these are the quality control steps, or really just data visualization steps of that pipeline. So if you go back to the graphic, these are the very first few cells. This says that we've basically got the same distribution of forward and reverse reads. From each sample, it tells you how many reads there are. Okay, so there's a sample here that has 0 reads. Okay, and and you might want to go, what happened? Never seen that. Anyway, there was a sample there with no reads in it. My QIIME viewer crashed. Don't know why. And so, right, I say this, I've said this often already this semester, it's important to look at your data. So you might want to just throw this one out from the get-go. Depends on whether you think that 0 is real, and you would go back to the metadata and say, okay, that, I don't know why. You could look that up in the metadata and see which sample was that on the table that I was showing before. Like, where was that collected from? Leaf litter or from feces, or what kind of sample was that? Oh, this is a fail, but that's okay. This is just the one. So this cell, I'm not sure why it's failing right now, but I created it to show that same QIIME viewer in the Colab notebook. I'm not going to take the time to fix it right now. I think actually I know how to fix it, but not going to worry about it because we just looked at it in the browser. So this is the step. Now data 2 is going to run. This is the one that takes some time because it's got to look at all the sequences, do the clustering, denoise the data, or error correct the data. And so what you can sort of get an idea from the names here what it's going to output. It's going to, the strain sequences, it's going to summarize those and then give us a table of that and then some stats just to describe quantitatively, how many different strains we have broken down by sample and stuff like that. Let me go over to the tutorial. So we're at this upstream data analysis step running data 2. And it explains each of these lines. This is also part of the quality control is trimming the reads. So if you remember way back to when we do our genome variant analysis and identifying SNPs, we used we used FastP, and then we used Trim-O-Matic this year. I forget which trimming software we used, but anyway, that's what Data 2 has, that trimmer built in, and we specifically are trimming nothing off of the beginning of the sequence, but then I think these might be 300 base pair reads that we're truncating them at 250, just because, like lots of Illumina data, you always get that pattern of high quality that declines at the end of the read. So this is just chopping off the last 50 base pairs of every read. And the rationale for that is in the tutorial. Mm-hmm. I didn't hear you. So the assignment for this week is going to be either to complete the single cell genomics and answer some questions about that, or complete this and answer some questions about this, and then the extra credit is if you do both. The data step or the one before that? Well, they're all bash. So this one errored. The start a local HTTP server. Is that the one that errored?
Mine's 9.
Yeah. What's the first? What's the QIIME? Actually.
The second one under uploading metadata.
Okay, so it looks like that finished. I've got my stats. And now I've got these two new files, the ASV QZA and table QZA. So the next couple are going to convert those into things that we can view. So it's going to tabulate them. Should go pretty fast. This is another thing. These cells give a lot of unnecessary output just on usage. But what you should see are no errors, and it's creating the QZV files that match those QZA files. The QZV ones are the ones that we'll look at in the viewer. And again, the rationale for all these steps is in the tutorial. So the stats table is giving us the sort of like whatever your trimmer might output in terms of how many it trimmed and stuff like that. So for example, percent of input that passed filter. So these are our sample labels on the left there. And you can see, like, for this sample, 86.9% of the reads passed the data to filtering for errors. It's sortable. So this is the one I was talking about before, that there aren't any samples in this one. So the fact that nothing passed filter is to be expected. One of the things that you would be concerned about potentially is if, okay, so now I have it sorted from the highest. So this was in some sense the cleanest sample, 95.5% passed filter. If that number was only like 50%, like, and that's your highest one, that's not very good sequencing. So there's something wrong. Okay. So I think anybody else having error issues? At least not until you get to where I have a note about making an error. So if you've run all the way down to the get the suboptimal classifier, you should have a pretty long list of files. And for most of them, you've got a QZA and a QZV matching. Just kind of. So as far as the homework goes, you'll complete the notebook, and I'll be specific about which steps need to be completed, and then, or maybe more specifically, steps that don't need to be completed. But then there'll be questions to answer about files like this and understanding, which the tutorial walks through. So this is the filtered, clustered table just by frequencies. And so, like, if you look in here at feature details, it's got these big gnarly names on them, but that's because what do you call this sequence that doesn't have a taxonomic ID yet? So the ASVs, right? They get these sort of unintelligible in some ways. I'm not sure if they're random. I'm not. Yeah, whatever the algorithm is that names these things, I don't know. But they correspond to a sequence cluster. So this is an ASV, and its frequency in our data, 1,334 times it saw that ASV, and it occurred in 23 samples. Okay. And so these are just graphical representations of that. These are sample IDs. This one has, right, this, you could think of this as an ordering of ASV clusters, right? So there are 1,218 different ASVs in that sample, and so on. Okay. Next step where I'm at is to get the classifier. And so this is the step that goes and takes those sequence clusters, those IDs, and tries to put taxonomic names on them. And so the output here is the taxonomy QZA, which, to look at it as a table, actually, I guess maybe we can look at the QZA. So without running it through the making it into QZV, just because we're running out of time here, you can see all those feature IDs now have taxonomic labels. Confidence is something like a blast score. I don't actually remember off the top of my head how QIIME computes the confidence, but it's scaled 0 to 1. 1 is very confident in that assignment. If you wanted to see what the sequences were that identify that, like if you wanted to take this, you could download this as a FASTA and do the blasting yourself. You've got your own database. Or you're interested in pulling out just the sequences for a certain kind of bacteria to see if it was in there. You could probably literally, well, go back to our BLAST module and exactly what we did with the beetle genomes. You could apply that to this FASTA file instead of a beetle FASTA file. Or ask ChatGPT to do it if you. Okay, so this you'll see in the tutorial. Boots does some pretty neat things, but it'll fail because it can't find Boots 2, and it's a problem that I could not overcome yet. I've tried all the channels. I have tried different ways of creating new environments to load on things that the Boots 2 developer says work. Could not get it to work in Colab. Oh, uh, to add insult to injury, not only does Boots 2 not install the outputs of, like how I said you might be able to skip, download, and just use that, these all go nowhere. So they can't make it work either. Yeah. The bottom one, the scatter plot, they have made it work in the past because you can take it over to viewer, and it is worthwhile. These are the results of if you could. So one of the things that Boots does is calculate those diversity indices that I talked about in the PowerPoint. So it suggests looking at the data in terms of these two. And this is one of the cool things of this provenance server. And it has all the metadata connected to the clustering analysis. And so this is clustered by the first two principal coordinates based on the Bray-Curtis, which is the actually what is Bray-Curtis telling us in this context of this tutorial? Not necessarily interpret this graph, but in general, what question is it answering? It's a beta diversity measure. And in my cartoon example in the PowerPoints, it was how different are the two forests in diversity, right? And so somebody translate that to what we're doing here. Yeah, I didn't spend enough time like talking about what these samples actually are. They're fecal and soil samples. I think there's actually a probiotic sample in here as well. But yes, it's how different are the microbial communities between the individual samples. Okay, that's what Bray-Curtis metric is telling us. And so if we color that by sample type, for example, this is my favorite thing about QIIME. Right, we can see that, all these red ones, these, actually I can't read that whole, the legend is cut off. That's not useful. I have to go back to the table of metadata to see. I forgot what that one is. But all these human excrement ones kind of cluster over here. So all of these things that you can color by are metadata that are attached to those samples. So here I've got it colored by the sample smell. Musty fecal. And these are the actual fecal samples, but these, what were those? They come from a bunch of different bulking materials, some food compost, soil nearby the toilet. Oh, this is the one that I was looking for a second ago, but because I can't see very well on on my computer. But this Sunmar microbe mix, I think that's the probiotic. That's this one. And the closest thing to it is food compost, right? So in terms of community structure. Anyway, some cool visualizations you can do there to try to understand what's going on in these environmental samples. You can imagine in a human microbiome study, each of those dots would be a patient, for example. And so you would be asking, what's the difference in diversity between patients? And we know that gut dysbiosis is super important in lots of chronic inflammatory bowel stuff. And so you might say, like, it wouldn't be surprising to see a cluster over here of individuals with celiacs or Crohn's separating out from normal based on their microbial community. So we are out of time. That's almost there. The collab keeps going. I think those will run once you've skipped past it, but you're gonna hit a wall at the end of the collab notebook. And so, you know, I encourage you as a bioinformatics problem, try to get through those, okay? Especially if you're in one of the labs that does metagenomics. And yeah, look forward to the assignment being posted this afternoon or early tomorrow.